as project collaborators. Yeah. So it limits for some of our divisions, some of the research. A lot of people commented about that. They don't have the capacity. They don't have the 4 collaborators to work with. I think we do. Okay, I think we do. So funding restrictions, members of a cluster may not hold alternative NRF research funding in 2026. If you, like me, you've applied for a wire-rated funding. If you awarded it, you must, if you won't be able to take it if you also apply for this. And then single discipline or individual applications are not eligible. It must be multidisciplinary. I had to stress there, I had to speak afterwards and mention that we are from a research center that is multidisciplinary in its nature, but within one division, within one department. They did not, so there were a few comments about that and they were not aware of it. So what they said in terms of concept notes, you will have to make it, it will have to be visible in your concept note that we are multidisciplinary, that there's different, it needs to be brought forward. They will also look at profiles more carefully, but the initial instructions, they had completely overlooked the fact that UCT's got this weird setup where they have research centers within divisions. So there's they're not going to support basic science, but they are saying that the executive pledged funding for it. They haven't put anything on paper yet, so it's coming. So, for example, people that are in astrophysics, astrophysics, etc. They were also in the room. They were also a lot of people were complaining. There will be opportunities for basic science coming up, but nothing solid has been created for them yet. But there is okay, so I think there'll be more than that. So concept notes, there'll be invitations for them in the next month. And if you miss this round, you'll have to wait another 2 years. For following year. Yeah. Any questions? Or do you want to clarify anything? No? Okay. So what they will be funding is what their priorities is human capacity development. There'll be a lot of points awarded to the societal impact of the work. What impact does your research have? So that will be the core. If you compare with basic scientists, they did acknowledge that basic science can't. Sorry, Alison. Basic science can't provide the same societal impact as a multidisciplinary research project. Okay, so if you have any questions, let me know. So next week we have Malcolm. He's going to talk more in depth about the genetic testing on musculoskeletal sports injuries theme, that the same talk he gave at the Health Fusion seminar, but in more depth. And this week we have Alison, the polygenic influence of genetic variants on ACL rupture risk across multiple international cohorts. Alison, the floor is yours.
Thank you for the invitation to share some of the students' work with you this morning. So I'm going to give you some insights of the research and our findings, but also some of the negotiations which happen when you're at a PI and how you have to present yourself in getting partners to work with you. And so Nancy has been very instrumental with me going to Australia to get our Australian partners on board. So how do we move this? Okay, there, I've got it. Thank you. So looking at musculoskeletal injuries across the globe, you can look at the purple on the map over there. Those are the the parts of the globe which has the highest burden of musculoskeletal injuries? Well, that's according to when they've taken the data using the ICD codes. But not everyone is using the ICD codes, and not everyone is efficient in using that. So that is just the map of those using the ICD codes, okay? What we do know that musculoskeletal injuries remain among the most leading causes of disability worldwide, and it's approximately 1.6 billion people affected globally. And this is from your studies. There's a whole lot of Lancet series that have been published. And they group the musculoskeletal injuries into 5 specific categories. For example, rheumatoid arthritis, osteoarthritis, low back pain, neck pain, as well as gout. So these are the data that they have included in these studies. I'm not sure if it is really neck pain or if it's lower back pain. It may be an error, but it's either neck pain or lower back pain as listed as the highest number of years living with disabilities, approximately affecting 70 million people globally. But musculoskeletal injuries are more than just those 5 conditions, okay? They represent more than 150 distinct conditions. And so they categorize all of those ICD codes into this category of other musculoskeletal conditions. And this range from acute injuries, chronic injuries that affect your locomotor, your connective tissue systems, such as bones, your joints, your ligaments, tendons, and muscles. And that's the area where we play in. And so it's always interesting to see what are the countries which have the most reported musculoskeletal conditions, and also where are those areas, geographical regions affecting the least. And so from an African, I'm busy writing a commentary paper. And so that's why I went to dig all this information, because as you can see now with NRF and most grant funders, they will see what is the priority areas. And so I want to make musculoskeletal injuries a priority area. And that's the thought process of writing the commentary. And you can see that women actually have the greatest burden of musculoskeletal injuries. More than 60% of the cases are actually women. And so women are disproportionately affected both by pain and disability. And if you look at the continent, Asia has the highest reported of individuals with years lived with disabilities, and Africa has the lowest numbers. And I don't think it's because we don't have it. We're just not reporting it. We don't have the measurements to record that data. And so that is very alarming. And so that is the mission of this commentary, is to say that, and Frederick is part of the authors together with Malcolm, is that we need funding to explore this. We need funding to look at Africa. And so musculoskeletal injuries has a huge impact on global health, and we require targeted interventions and research to address the growing burden of these family of diseases. And so the area where I play in is genomics. And so I want to apply genomics to musculoskeletal injuries and to see, can we identify using genetics and as well as the omics approaches to treat, prevent as well. And so if you look at genomics in performance, the human genome was mapped, started in 1990, and we had this map in 2003. And then people had said, oh, now that we've got the human genome, we're going to be able to cure every single condition. We're only now starting to understand some of these proteins encoded by these genes. And we're so far away from understanding all the sequences within our genome. So we may understand some of them, but the large majority, we have no idea what is the functional significance of those variants which are sitting within the intronic regions, which do not make up our protein. And so we had this boom of sports genomics in 2008 to 2012. And at the beginning, the focus was, can we identify the genes causing elite athletes? Can we identify talent? And that was the main focus. But now we've we've matured in science as biologists, and now we're interested in what sets two people apart? Why, if we have two people having the same exposure levels, but yet only one receives the injury, what is the driving factors controlling that? So can we use genomics to understand that? And more important as well, can we use genomics to understand not only performance enhancement, but also what are the factors governing recovery? Okay. And so there's a shift in using genetics to treat and to prevent. And so looking at ACL ruptures, there's much evidence to show that there is a familial component to sustaining an ACL rupture, and it is said that it is approximately 70% of ACL ruptures have a heritable component. Doesn't mean 70% sounds quite large, but it doesn't mean that the genetics is a major contributor to that risk profile. It just means that they found sufficient evidence to showcase that there is a heritable component to that your genes do play a role. So if you look at height has roughly a heritability component of 80%. So it just means that there's a large component where we can contribute genetics to, but doesn't mean it's the major component. So you just have to wrap your mind around that. And some of the genes that were implicated in the beginning are our collagen encoding genes, our enzymes which regulate substrates within your extracellular matrix, as well as your pro-inflammatory genes, as well as our genes that upregulate your angiogenesis, which is the formation of new blood vessels. And so we're also trying to see how these genetic information can also make us understand what is the intersection between aging. If I'm getting closer to that category of aging, when you're getting up in the morning and everything is stiff, right? So how can we take the lessons learned from these sporting injuries to understand what are some of these joint issues that we're experiencing as we age as well? And so much research was put into place to showcase that actually there is a heritability component and there is a family affair. So when Nancy and I went to Australia a good couple of years ago, and we were the only two females in a room of about 20 scientists and largely orthopedic surgeons, trying to convince them that there is a genetic link to ACL ruptures, and trying to convince them that we were the lab to do this research. Do you remember, Nancy? We were put on the spot. And the one professor who was in orthopedics, big orthopedic surgeon. What's his name now again? I forget his name. Anyway, so he said, well, actually, when I see a patient coming in for an ACL repair, I'm bound to see a first-degree relative also coming in with an ACL repair. So there's evidence to suggest that there's a twofold increased risk for first-degree relative to sustain an ACL repair if they have someone already with an ACL rupture. Okay, and so Malcolm started this research looking at the genetics of soft tissue injuries, starting with Achilles tendinopathy and then later into ACL ruptures, with Mike Posthumus starting that research, being the first student on there. And so why do we choose ACL ruptures? Well, ACL ruptures of the ACL is not the most prevalent injury of the knee, but it's a significant proportion of the injuries to the knee involves your anterior cruciate ligament. And what's significant is that it actually affects those college players, young adolescents, and athletes who are like in a 20 to 28. And so that numbers are increasing. And so we are going to have a proportion of individuals, because if you have an ACL rupture, your incidence of early onset osteoarthritis increases, and it's approximately 40 to 50% of those individuals will get osteoarthritis within 10 to 15 years after sustaining an ACL rupture and an ACL reconstruction. So we're going to have a population, those numbers are steadily increasing, a population with high numbers of osteoarthritis and high numbers of reduced mobility and quality of life being affected. Also, having an ACL rupture has a huge economic burden on governments, and not only that, also the not only the surgery, but also the what am I looking for? The systems to offer rehabilitation is also significant. And so we started looking at this research, and we're leading in the world, and there's about more than 80 loci. What are loci? Specific spots on the genome that have been implicated with either an increased risk or a decreased risk of sustaining this ACL ruptures. And we looked at structural components. The major structural components found in ligaments are your collagen genes. And so we have several loci, specific spots on these collagen genes which were linked with risk. We also have proteoglycan. The proteoglycan family is my favorite proteins. These proteoglycans encode for proteins which can also contribute to the building blocks of our tendons and ligaments, and so they act as structural components, but they also act as signaling factors, so they can also upregulate downstream processes such as inflammation, such as angiogenesis. Okay, so that's why I like these proteoglycans, because they're quite diverse. Then you can also get genes which code for enzymes which regulate the components within our extracellular matrix. And then you get other genes which code for proteins which have to stop these MMPs. Okay, so there's always this balance of maintaining the homeostasis within our tendons and ligaments. And so there's a whole host of genes coding for proteins, which either upregulate some proteins, and you have other genes coding for proteins which downregulate them, depending on the condition that is required at that time for your connective tissue. We also found loci within our apoptotic genes, such as your caspases. Apoptosis plays a very important role in maintaining the tissue integrity of tendons and ligaments. And of course, cytokines. Cytokines, though, for example, a condition as chronic Achilles tendinopathy, it's a chronic condition. It is not an inflammatory condition, but these cytokines, which are pro-inflammatory, activate the healing pathway. Okay, so they will upregulate apoptosis, they can upregulate angiogenesis, they can upregulate different collagens from being expressed as well. And so all of these genes play together and they fit together to get an optimal functioning connective tissue. So I've spoke about these loci that we've implicated within genes, but what are these loci? I'm actually referring to a genetic variant. So these genetic variants are common amongst all of us. You and I both share these genetic variants. So what it is that at a particular site, you may have a different spelling. So you may have a C, I may have a G, I may be homozygous for the CC, that is that both my strands are CC, or I could be homozygous for GG, or you could be heterozygous. You could have a C and a G. And so in the lab, we track and count of our controls, how many of them are homozygous CC or homozygous GG, how many of our cases, what profile of that particular variant do they have? And then statistically we say, well, actually, our controls have this particular genetic profile, and it's more often seen in our controls, and there we can say, well, therefore, that particular variant is more associated with a decreased risk if it's within our controls because they don't have an ACL rupture. Or if it's more prevalent in our cases, we'll say, well, actually, it looks like that particular variant is associated with an increased risk. Let's look at the protein. How does that variant impact the expression of that protein, and how does that protein function within our ligament? And so that's what we do continuously. So we can look at this genetic variant. What is the impact on the protein? not necessarily the function of the protein, but how much protein is produced. Is there an increase in expression or is there a decrease expression? And so these proteins are all expressed. Remember, our nucleus is within our cells. And the cells, I always say, is the brain of every single tissue. The cells will regulate and control the expression profile of all the proteins required for the structural integrity of our tissue and also for the maintaining homeostasis of our tissue. So, depending on the population of proteins and how much proteins are produced, your tissue will have an optimal function. Okay. And that tissue makes up your organ and will dictate how your knee will respond to loading or unloading. These are 3 papers which I've used in preparing the study, and it all came from our research team. And so please go find them. So the first paper I want to draw your attention to is the paper with our past PhD graduate, Andrew Gibbon. And in her study, she looked at whole exome sequencing for Achilles tendinopathy. And then years later, we had a PhD graduate, Danielle Feldman, and she conducted a whole genome sequencing study using a twin approach. And both these studies were the first to be presented in the world. And then Senator Dlamini, who's actually a Swazi princess, true story. And she's busy writing up a PhD. I'm supposed to be reading her discussion chapter now. And so she used some AI technology before AI became big buzz. But we decided, there's so much data out there in public domains. Let's take all the data that we have connected to tendon and ligament injuries, and let's come up with a study design where we can see, well, what are all the proteins and what are the genes coding for those proteins which have been implicated in a phenotype linked with ligament injury or a phenotype linked with tendon injury. So she's developed this biomedical knowledge graph, which we're now using to decide what genes should we look at. But more important, we're also using the information that Andrea got from her whole exome sequencing study. For example, I remember presenting for any ethics proposal, we have to present your data before it can be submitted. And so I presented this whole exome sequencing study, and I said, no, Alison, that's not going to go anywhere. That project has too many risks. It's a high risk project. I said, oh, that's fantastic. I like high risk projects. So our thought process was, we only had like a couple of R100. We didn't really have a dedicated fund to support this whole exome sequencing study. And from a cost perspective, then it was about 10,000 Rand to do an exome sequencing, to sequence an individual's exome. And we didn't have a dedicated fund. I don't know where I got the money from, but we got the money. So we sequenced 10 cases. And these are what we call exemplar cases. They had bilateral tendinopathy injuries. And we sequenced 10 controls. And so they were much older than our cases. They were physically active. So the thought process they were exposed to more risk factors, more loading, yet they were protected in a sense, right? And they had no previous ligament or tendon injuries. And so we sent it away to a company in Hong Kong, or was it the States? I think it was the States. Autogenetics is the States. And then we came back and we had millions of sequences. We didn't know where to start. We sat for days just sitting, literally just sitting. How are we going to analyze all the sequences? Because we only had 10 cases and 10 controls. So I said, I've got an idea. We know that these sequences are found in all of us. But what we are interested in is, what are the most unique sequences in our cases, and what are the more unique sequences in our controls? Yes, that made sense. So I said, can we therefore say, let's look at ARB number of 30%. If we look at the frequencies of all these sequence variances that we've identified between our cases and controls, let's draw a line and see what is the 30% threshold difference of these sequences between our cases and controls. So now we've got a pool of sequences which is more common in our cases only in these 10 samples or 20 samples, and they're more common in our case, in our controls. So these are just some of the genes that we've listed where we know that there's a 30% frequency difference of specific polyvariants that we've identified from the sequencing, only found in our exons. Our exons are the coding bits which are used to make our protein. So it's not looking at any intronic sequences or regulatory sequences. And so we started exploring that. So now we've got that information of these potential variants which we could use in our other studies. But then, okay, we did the exome sequencing, we've ticked that box, but let's do some whole genome sequencing. That's where things became sexy. So when we were in Australia, I said, oh, well, now we've got your DNA samples. Do you think you can look at your databases, your patient data? Are there any are there any families, or do you have any notes on individuals who were actually twins who ruptured their ACL? And I said, Alison, no, I'm not thinking about that. We're not going to do that. No, that door was just shut. I tried a couple of times and I said, no. Then I spoke to, it was some conference happening here, and I spoke to John Patricia. I said, John, my next sexy thing is, can we look at families where they had twins and both twins sustained an ACL rupture? And he was absolutely fabulous. I'm on it, Alison. And then he found two families for me who had twins in each of the families who sustained an ACL rupture while they were at school. And so from a genetic perspective, particularly for a multifactorial condition, such as a sporting injury, you want to look at a profile where the individual sustained their injury very young. So that will indicate that potentially your genetic component is more than just people who sustained an ACL rupture at the normal age of 30 and above. Okay, so that is one indication. And so we found these two sets of families, and we did a whole genome sequencing on on everyone we could get bloods and exact DNA from. And we looked at what are the sequences which are unique to our cases. So we got a whole list of that. We got what are the sequences unique to each family. We also did a full screen to see, are those 80 loci that we previously sequenced or implicated, how many of them are actually found within these two family sets, and how common are those potential risk-associated loci found in, because there's not so much data out there on whole genome sequencing data there. If we chuck that onto the whole genome sequencing, which is available on the public domains, how many of those individuals actually have those sequence variances that we've identified? And we found that these two families had the highest burden of those variants compared to published data of just random controls. And so that's showing us that potentially those variants that we're finding in those 80 loci are maybe real, and that it's not just a fluke by chance that we found those associations. So we're still trying to unpack that. So what did this study show us? This study showed us not only particular loci with particular chromosomal regions that contain potential structural signatures or maybe regulative signatures, which we could use to now explore in our ACL rupture phenotypes, but also highlighted specific pathways. For example, our collagen genes. It highlighted genes involved in promoting the angiogenesis, etc., as well as proteoglycan families. And so we went back to Senna's study, knowing that we're looking at this 30% difference. Senna highlighted, she looked at all the public domains. She looked at phenotype descriptives which were associated with ligament or tendon phenotype-related injuries or anything related to tendon and ligament. Then she saw how often do those genes map to other known databases. So we found 2 or more links. So it has to be, so that was our evidence that there's strong evidence to highlight that particular gene is associated with a ligament phenotype. So we had about just under 3,500 candidate genes to explore. Then we took that information from the whole exome sequencing and particularly that 30% differences, and we found 411. And so now we're systematically going through screening those genes. Particularly interest was this HSBG2. I forget what it stands for. Please forgive me. But the protein is, hey, it's the proteoglycan name is perlecan. So that's what I remember in my brain. And then integrin beta 2 encodes for an integrin beta component, and then FGF9, which stands for a fibril growth factor 9. Thank you, Nancy. And so we thought maybe we can find a missing heritability looking at these genes. And so what we find is that your integrins, so when I teach the first year students as well as our honor students, I say that the cell is the most important part in our tissue. And then cell-to-cell communication is pivotal. So our tendons and ligaments have a low density of cells. So there's a very small proportion of cells relative to the extracellular matrix. For example, in muscle tissue, your cells is the major component in that tissue, and your extracellular matrix, that is the environment outside of the cell, is your minor component. But in tendons and ligaments, your extracellular matrix is the major component physically, and the cells, there's very few cells in our tendons and ligament. However, the cells are responsible for making all the proteins needed to function within our connective tissue in tendons and ligaments. And so that cell shape will send a specific signal to say, if the cell shape changes, that is a signal, whatever signal it is, to say, oh, I need to produce, we're under attack, or we need to do remodeling, and so I need to produce this profile of proteins, okay? Cell density has a similar signal. If the cell density decreases, that'll indicate to the cells, I'm under threat, I need to produce more collagens, or I need to produce more MMPs, etc., etc. Or what will say, actually, let me increase my cells because I need to do a lot. I need to produce so much more proteins. And so you find there'll be areas within our tendons and ligaments that there's an increase in cell density, and other areas will be a decrease in cell density. Because the cells is the brain. They're producing everything that is needed to maintain that structural integrity of our ligaments. And so looking at the integrins, they form these integrin complexes with the head sitting into the extracellular matrix. It has the body which sits embedded in the cell membrane, and it has a tail which sits within your cytoplasm. And so the head is able to engage with molecules such as your different substrates, such in this case, it's your collagens. And with that molecule binding to your integrin complex, it will submit a signal to the inside of the cell. Similarly, the inside of the cell will also submit a signal via these integrin complexes to engage with other substrates. Okay, so the integrin family of proteins, and we looked at a specific integrin gene, and we looked at this particular polymorphism. How much time do I have? Okay, I'm almost done. So this particular variant doesn't change the amino acid sequence, so it doesn't change the coding of our protein. But what we found when we did protein dynamic modeling, which took a couple of months because you need so much computer power to generate this modeling, that it changes the fold, it changes the structure of the protein. Can you see? If you got a C over here and A, it looks very different to if you have the T allele on B, and those what's the color? Green color. Those are your helices, and then your purple colors is your beta pleated sheets. And so our protein is more open. Maybe it's more open and ready to mingle with other extracellular matrix proteins. You have a T, where the C is a little bit more compact, right? It's choosy who's going to mingle with, which substrate is going to choose. And so we found that if you have the C allele at the integrin beta 2 locus, you have a decreased risk of sustaining an ACL rupture. If you have a T, you have an increased risk. And so part of Lataba's master's project, she looked at integrin beta 3 because, as I showed you earlier, that is integrins form multiple complexes with other integrin molecules. It's not just one integrin molecule. And so she looked at integrin beta 3. Lataba looked at HSBG2 Perlican, but only in the South African population. So we wanted to look at across the cohorts that we have. So we had cohorts from Sweden, Poland, South Africa, and Australia. And so finding a genetic variation, you want to look at it in multiple populations, because if you want to identify biological significance, you have to do it across the globe. You can't just be looking at a South African, because is that truly biologically significant, or is that just here on the tip of Cape Town, right? We're playing in the global sphere, so we want to find something which has, and doesn't mean that it has biological significance, that it has clinical significance either. You have to show and prove that still. So we looked at the sperlican, which is a proteoglycan. Remember I said previously, proteoglycans have a structural role. So they can either bind to your collagen fibril, or they can embed between your collagen fibrils, and they can have a cell signaling role. So they can upregulate different pathways. Integrin and B3 and integrin beta-2, they code for particular subunits of this integrin protein complex. And so they facilitate cell-to-cell communication, they facilitate cell matrix communication, and they facilitate mechanical, they're able to sense mechanical loading on your connective tissue and activate repair processes. So Perlican actually is able to sit between a collagen as well as elastic fibrils. And so they're almost like this reinforcer. And your integrins, particularly integrin beta-2, is also able to bind to actin. And so by binding to actin, they produce a force within the cell, which is transmitted to the outside. And that force not also able to change the shape of your protein, but it also dictates the proteins which are able to bind to your integrin beta complexes. Okay. And so that force can also change the shape of that protein being able to bind to it. And so that mechanotransduction plays a critical role in the tissue. And maybe that's why it doesn't matter maybe that explains why the CC genotype that we're seeing is so dominant in predicting if you have a reduced risk or an increased risk. I'm thinking and hypothesizing. So we looked at all our cases and controls, just under 1,500 samples. And what did we find? That if you have the, can't see this, is that an AG? If you have an, if you have an AG over there, if you look on my figure, the graph, you have the genotypes on the x-axis, and then you have the frequency of the genotypes on the y-axis. And there we see that you have an AG that's more often found with our controls. So the AG genotype is associated for Perlican is associated with a decreased risk. And in fact, there's about a 35% reduced risk of sustaining an ACL rupture if you have that particular genotype in the Perlican gene. We also looked at haplotype analysis. That means a combination of these genetic variants. And we found that if you have a GA, you also have a reduced risk of sustaining an ACL rupture. Then we looked at integrin beta-3. Integrin beta-3, we found if you have a C, you are at this particular locus, you are at an increased risk. But I showed you that these proteins don't function on their own. They function in together. They have an interdependent relationship sometimes. And so we did gene mania analysis, and we found that they're all connected. So these different lines show you how strongly they're connected. If you have more than one line, the connection is stronger via different pathways. Okay. And so we looked at integrin beta-3, HSBG2, as well as integrin beta-2 together. So we looked at all the potential combinations between integrin beta-3 and integrin beta-2, and you found Doesn't matter what you inherit at your integrin beta 3 locus. If you have a T at the integrin beta 2, you're going to be at an increased risk. And though you can see that the effect sizes are quite small with odds ratios just over 1. However, we're looking at multifactorial conditions. We're also looking at more than one gene. So these effect sizes are biologically relevant still. But let's look at all 3 genes. So we put all of the information together, and we also did multiple correction for multiple testing. So whatever p-value we're getting now is quite significant. And we found that if you have a GA, remember GA previously was associated with, what was it, increase or decrease risk, but that whatever that GA haplotype was for HSPG2, it's holding true now. And if you have the A at integrin beta 3 and the C at integrin beta 2, you are at a reduced risk of sustaining an ACL rupture. Nearly 40% reduced risk. And so we've shown that studying 1,500 cases and controls, that the HSPG2 locus is potentially significant. We've mapped it to a specific region on this gene by looking at the haplotype. We've shown that if you're only looking at integrin beta 3 and integrin beta 2, and you know they form a complex, doesn't matter what you get, the T is the dominant, and you're going to be at risk. We've also looked at all 4, and we show that having a specific allele combination, at least you have a moderate protective effect, 40% reduced odds of ACL rupture. And so I have explained to you how HSPG2 functions in stabilizing your collagen matrix and how these integrin families of proteins regulate forces which regulate cell shape as well as particular proteins binding to it. And so our next step is that's what Nancy's work is, to look at functionally, can we come up with a model to show that if you have these particular genetic profiles, how does that function your protein expression? What we have found that these particular variants that we've looked at, they actually coincide with expressed quantitative trait loci. So that means that these variants are associated with either more RNA being produced or more protein being produced. And so we're trying to sit with that data, see which tissues are affected, and what particular genotype is associated with an increased protein or decreased protein, and then how can we look at it together. And so the proteins are your main, is the brains. It regulates and controls tendon remodeling and ligaments remodeling, how we adapt and how we repair. And so I'd like to acknowledge the James team, and the funding for the studies are from the South African Medical Research Council. We have many collaborators across the world, from Poland and Sweden and Australia. Thank you.